So enzymes decrease the activation energy for a specific chemical reaction. Again, breaking down of fats is one example of that. So it's going to decrease the amount of energy we need in order to break those fats down to their smaller components, triglycerides, right? And when we talk about the activation energy, does anyone know what that means? Or another way to say that? What is the activation energy? Yep, so the specific amount, more specifically, the least amount of energy that would be required. So what is the least amount of energy that it would take the body to proceed with this reaction? And enzymes lower that activation energy. So they make it so that even with less energy than in the absence of an enzyme, we can proceed with this reaction much more faster, okay? So they bind to a reactant, also called a substrate here. We have the enzyme binding to the substrate on the left, and then we have this bidirectional arrow which shows that this reaction can proceed in the forward direction as well as in the reverse direction. We end up with an enzyme substrate complex here in the middle, and then we spit out the product on one end, and then we can spit back out the enzyme as well, okay? Now this can proceed in the reverse direction as well. So we can bind the product that we made to the enzyme. We'll eventually end up with an enzyme substrate complex, and on this end, we can spit out the substrate once again, and we can spit out the enzyme as well. Okay, so this can happen both in the forward and the reverse direction, depending on the environment in our body. So if we have lots of glucose, we're going to store it up into glycogen. If we have lots of glycogen, we're going to release it as the body needs it. So these enzymes are kind of sitting in the middle, catalyzing the reaction forward, catalyzing the reaction backward, depending on the substrate concentration and the product concentration on either side. Okay, so let's illustrate how enzymes actually kind of work. Okay, so here we're speaking about the activation energy barrier. Okay, and we're going to draw this graphically. So we're going to draw a graph. On the y-axis, we have time in seconds. On the x-axis, we have energy needs in kilocalories. So this is how much energy we require in order for this reaction to proceed. Okay. Now we're going to draw two lines on this graph. One is going to represent the reaction proceeding with the enzyme. And one is going to represent the reaction proceeding in the absence of the enzyme, so without the enzyme. So without the enzyme, the reaction proceeds, it takes this amount of time, that amount of energy, and then we get to products. So on this side, we started with reactants, okay, or substrates. Now with the enzyme present, again, we're starting with reactants or substrates, and then we need less energy, and we take much less time to get to products. Okay, so as you can see here, the enzyme lowers the activation energy that is required for this reaction. All right, so this is the activation energy. All right, so that's the first thing that the enzyme does. Right, it lowers the activation energy. So I'll put that over here. The second thing that we can see is that the reaction proceeds much faster with the enzyme than it does without the enzyme. Okay, and that's the second thing an enzyme does, is it speeds up speeds up the rate of the reaction. Right? So enzymes lower the energy. We need less energy for reaction to proceed in the presence of an enzyme, as well as make the reaction go faster so we get to our products and vice versa. Right? Remember, this can happen in both the forward and the reverse direction in much less time than it takes in the absence of the enzyme. Okay. All right. Any questions on this before we move on? Any questions? Okay. So let's continue on here. So this graph is depicting just what we saw, right? The lowering of the activation energy and the speeding up of our reaction. Now we're going to move on to speak about the substrate specificity. And so basically we ask ourselves the question, if an enzyme can speed up the rate of a reaction, why don't enzymes just walk around the body and speed everything up, right? Why do we have very specific binding of one enzyme to one substrate? For example, lipase only binds fats, whereas trypsin only binds proteins, amylase only binds carbohydrates. How do we get that specificity where an enzyme can only speed up one type of reaction by binding to one type of substrate? And to answer that question, we have two different models, two different theories to answer that question. The first one is the lock and key model. Now, the lock and key model says that an enzyme has a very unique activation site. And basically, the shape of the activation site perfectly matches the shape of its unique substrate. So basically, only one type of substrate can bind to this enzyme at its unique activation site. Now, the problem with this first theory is that it does not explain the reversibility of these enzymatic reactions. So why can the reaction proceed in the forward direction, right? Substrate binding to enzyme, and then we get a product. And if that product is not specifically shaped the way the substrate is, how do we get that reaction to proceed in the reverse direction? So this is a problem with this first theory, right? It does not explain the reversibility of enzymatic reactions. The second theory is a little bit better at that, right? It is the induced fit model. And basically what we say in this second theory is that a substrate will get close to an enzyme and then it will induce a perfect fit. So initially the active site looked a little bit different than the substrate, but when the substrate sort of affiliated itself with the enzyme, we induce a perfect fit so it matches the shape of the enzyme's active site. So basically it's not this lock and key phenomenon, but it's more of an induced fit. So I can make the enzyme shaped the way the substrate is, and we can induce a perfect fit that way. So we have the reaction proceeding in the forward direction, we get a product, and the product is again differently shaped from the substrate. But when the product interacts with the enzyme, the product can induce the enzyme to kind of fit it, and then you can have the reaction proceeding in the reverse. So the induced fit model is a little bit better at explaining how we can have reactions proceeding forward and reverse. Okay. All right. The next thing we'll talk about here are cofactors and coenzymes. So these are important functional molecules that are required for enzymatic reactions. Cofactors are basically non-protein components of some enzymes, and these are necessary for the enzyme to hold its function or to hold its conformation, right? Its functional conformation. These can be trace metals like magnesium, copper, zinc, or iron. And they're usually sort of just supporting structures, just helping along the way. Whereas coenzymes are non-catalytic molecules, but they are necessary for the proper functioning of the enzyme. These are usually derived from vitamins, and they basically can help by transferring chemical groups from one molecule in the reaction to another molecule. So to kind of simplify this, the way I like to think about this is, think about driving a car. Think about the headrest as being a cofactor, right? The head seat behind you, whereas the steering wheel is more of a coenzyme. So we can drive a car without a headrest, but it's much more comfortable, and the headrest provides some support and kind of helps the process. but we cannot drive a car without a steering wheel, which is a coenzyme, okay? And so the coenzyme is much more important for the functioning of the enzyme and is necessary for the enzymatic reaction, whereas the cofactor is just sort of a supporting structure that kind of helps along the way, right? I also want to point out here that coenzymes are not necessarily catalytic, meaning that they do not themselves speed up the reaction. They just kind of work with the enzyme. And again, going back to that analogy, think about driving that car, right? The steering wheel is important for driving, but the steering wheel is not you, right? The steering wheel is not actually driving the process, but it's really, really important for driving the process. Okay, and then let's talk about some of the enzyme reaction rates. So an enzyme can be affected by, or the rate rather, can be affected by a couple different components here or factors here. So the enzyme's catalytic rate depends upon how many molecules of product or substrate, remember this can happen in both directions, are generated in time. So this is a rate. So how fast is this enzyme working? And that depends on the substrate concentration and the enzyme concentration. And these first two factors kind of make sense, right? If we have more substrate, we're going to have more of a reaction happening faster. If we have more enzyme, we're also going to have more of the reaction happening faster. We have more stuff around, so it's going to proceed much faster than if we have less substrate or less enzyme. The third factor here is the affinity of the enzyme for the substrate. Now, the affinity really talks about how much do the substrate and the enzyme kind of like each other. How willing are they to bind to each other? And that can vary depending on a lot of different factors, like substrate concentration, enzyme concentration, the environment, the local environment, like the pH and temperature. But basically, how willing or how affilious are the enzyme and substrate to binding to one another? If they have a strong affiliation, we have a faster rate. If they have a weak affiliation, we have a slower rate. And then the last factor here are temperature and pH. And this kind of makes sense. This ties right back to the fact that enzymes are proteins, right? Enzymes are proteins, and so they're going to be denatured and be dysfunctional at excess temperatures and at acidic or alkaline pH. So this ties right back to homeostasis. The body needs to be in a consistent, constant, internal environment in order for things to kind of work properly. And enzymes are one of those examples. So if we have a high temperature, we may see enzymes moving much faster. If we have a high pH or a low pH, we may see enzymes moving faster or even being denatured if that's